I'm Rochelle Crosby-Watson, and I'm a professor in integrative biology and physiology. And today we're going to learn about skeletal muscle physiology. So let's get started with a case study. A neurologist has several patients in her practice, and they're presenting with inherited signs of muscular dystrophy. These symptoms are all very similar to one another. So she gets biopsies and sends them off to a neuropathologist for analysis. The neuropathologist then conducts electron microscopy, and the images are shown here. So we're looking at striated skeletal muscle. What do you think about these images? Do they look normal to you? Can you label the structures? Do you see anything that might interfere with the function of skeletal muscle? Well, in today's lesson, at the end, you will be able to tell us a lot about these skeletal muscle biopsies. So, there are 3 different types of muscle. Broadly speaking, there's skeletal muscle, cardiac muscle, and smooth muscle, and they have different properties. Let's take a closer look. So smooth muscle cells are individual muscle cells that act as a syncytium. That means that the smooth muscles can communicate with one another by virtue of structures called gap junctions. What's also distinct about smooth muscles is that they're much more irregular in patterning. The nucleus, as you can see in this diagram, is actually located in the center of the cell, like it is for a lot of different cells. That's not the case for skeletal muscles, you'll see in a moment. So smooth muscle can be found in many different organs, including blood vessels and the intestine. So smooth muscle really is everywhere. Cardiac muscle is one of 2 different types of striated muscles. Cardiac muscle, like smooth muscle, is involuntary. The nucleus is also at the center of the cardiac muscle fiber. What also makes cardiac muscle similar to skeletal muscle are these distinct striations that occur inside the cardiac muscle. The cardiac muscle, like the smooth muscle, is able to act as a functional syncytium. So there are structures in cardiac muscle, the intercalated discs and gap junctions, that allow one cardiac muscle cell to interact with another cardiac muscle cell and to communicate with each other, allowing the cells to regulate and time their function, which obviously is very important for the functioning of muscle in general. So skeletal muscle is formed from the fusion of many different muscle cells. And so skeletal muscle fibers are quite long and large. The nucleus of the skeletal muscle is not normally at the middle of the cell, and in fact, it's at the cell periphery, which you can see in this diagram. And the skeletal muscle cell is packed with proteins that contribute to the function of the skeletal muscle to contract. And those proteins form these really distinctive, gorgeous striations. You're seeing them here in an illustration, but we will see real examples of that by electron microscopy. So skeletal muscle is going to be the focus of our course. We will talk a little bit about the function of cardiac muscle and smooth muscle as it relates to disease, but we're going to be mostly exploring skeletal muscle. So the introduction will focus on that. Skeletal muscles are found throughout the body, as you know. They're connected to bone, which they move by virtue of a tendon, shown here. So the organization of skeletal muscle is very layered, and those layers allow for the regulation of skeletal muscle function, which is to contract. The whole skeletal muscle is divided up into individual skeletal muscle fibers, also called myofibers. Those myofibers can really vary in both diameter and in length, depending on the size of the person in the case of patients that we're going to be talking about in our course. Each individual myofiber or muscle fiber, which is the cell, is comprised of these very distinctive cylindrical structures called myofibrils. And it's the myofibrils that do the work of skeletal muscle. So the myofibrils make up 80% of the muscle volume, and they're about 1 micron in diameter, and they run the entire length of the muscle cell. And lastly, those myofibrils, as we'll see, are composed of things called thick and thin filaments. These are structures that are very organized within the cell, and they comprise actin and myosin, as we're going to see, and it's those interactions that really regulate and drive skeletal muscle force production in a very elegant fashion. So the thick filaments are called thick filaments because they're much larger than the thin filaments, and the diameters are shown for you here. So let's get back to the connective tissue layers in skeletal muscle. The connective tissue layers are really a critical part of the structure of the overall muscle. And as we're going to learn later, when we start to really get into the mechanisms of disease, the connective tissue interaction with the skeletal muscle fiber is really critical for the function of the fiber. So I hope that you can see in this diagram that the skeletal muscle is connected to the tendon, and that outer layer of connective tissue is called the epimysium. There are other layers of connective tissue, including the paramecium and the endomysium, okay? And we're going to see those, I think it's a little more clear on the next diagram. So if we were to take a transverse cross-section of skeletal muscle, we'll see that that outermost layer of connective tissue is called the epimysium. So think of the epimysium as the connective tissue that's wrapping around the entire skeletal muscle. The endomysium is the connective tissue layer that wraps around each individual myofiber. Remember that the myofiber is the muscle cell. So that endomysium is actually really a critical component of many forms of muscular dystrophy. So also, if you take a look at a whole skeletal muscle cross-section, you'll find that the groups of skeletal muscle fibers are separated by connective tissue layering, and that's called the paramecium. So the paramecium wraps around groups of individual skeletal muscle fibers, and those skeletal muscle fibers, when they're grouped together and separated by a paramecium, are called the fascicles. Okay, so that's your basic dissection, if you will, of the connective tissue layers in skeletal muscle. Now, what sometimes can make science and research confusing is that we have many names to indicate the same structure. And I just want to point that out to you now. And as scientists, we flip-flop back and forth between all of these. So a skeletal muscle cell is also known as a muscle fiber, a myocyte, or a myofiber. Any of those terms are correct. Okay, so based on what you've seen so far, number in order from the smallest to the largest the different components of skeletal muscle. Think about it for a moment. So the answer is here. The nucleus is the smallest component. It's inside the cell. Remember that the nucleus is located at the cell periphery in skeletal muscle. It's not normally located inside the cell. And what we're going to learn later on is that if we do find that the nucleus is located in the middle of the cell, that's a sign that something has gone wrong. So the nucleus is the smallest structure in this particular listing. And next is the myofibril, which is inside the muscle fiber. Then the skeletal muscle fiber itself, the fascicles, and finally the skeletal muscle itself. The next question. Number in order from innermost to outermost the layers in skeletal muscle. Here's the answer. So number one is the sarcolemma. The sarcolemma is the membrane that encases each skeletal muscle fiber. It sits just underneath the endomysium. After that, the paramecium is a connective tissue that will segregate groups of skeletal muscle fibers, also known as fascicles. And finally, there's the epimysium. We're going to look at the sarcolemma in more detail. Take a look at this great illustration from Pearson Education. This is an oblique view of skeletal muscle, and I hope that you can see the paramecium, which is grouping together the fascicles in the inset. I really like this as an image. It shows you the whole structure of skeletal muscle. It's also showing you these wonderful layers of connective tissue that we've been talking about. And the layers of connective tissue are really actually very important to the overall function of skeletal muscle. They provide elasticity and flexibility, but they also provide protection. The skeletal muscle interacts with the connective tissue at all layers, as you can see. The analogy that I like to use is a steel-belted radial tire. So the rubber itself is pretty flexible, but the steel-belted radials give structure to the tire. Another analogy that you could think about is a snail with its shell. The snail itself is very flexible and pliable, and without its shell, it would be quite susceptible to damage. So Keep these analogies in mind when we start to talk about muscular dystrophies. This theme is going to come up again, and we will see how this loss of connection between a skeletal muscle fiber and its extracellular matrix can contribute very significantly to disease and the inability of skeletal muscle to do its function. Okay, we're going to look at skeletal muscle in transverse sections and longitudinal sections. And what you will come to appreciate is how we can get different information by looking at skeletal muscle from these different angles. So a transverse cryosection is something that looks like this, where we're cutting the skeletal muscle in half and we're taking a look at the skeletal muscle in a transverse orientation. So if we do that and then stain skeletal muscle using histological reagents and view the skeletal muscle under electron microscopy, we see a really beautiful striated pattern, okay? And just to orient yourself, what we're looking at in this particular image is one single myofibril that was isolated from one muscle fiber. And again, you can see these gorgeous repeated patterns. At the inset, I hope that you can see that there's a repeated pattern of thin filaments and thick filaments, okay? And in fact, every single thick filament is surrounded by 6 thin filaments. And this structure repeats itself throughout the entire skeletal muscle fiber. Now, if we take a longitudinal section, shown here, and view the skeletal muscle from this longitudinal angle, we have a very different picture. If we look by light microscopy, which is at fairly low magnification, I hope you'll see again that we're having these gorgeous this repeated pattern, these striations, that the nucleus is located at the periphery of the cell, right underneath the sarcolemma. Each of these skeletal muscle fibers are surrounded by a plasma membrane. That plasma membrane is called the sarcolemma. The thick and the thin filaments is what is creating the structure, that striated structure that you can see so beautifully by light microscopy. Now, if we take a closer look by electron microscopy, which gives us a higher magnification view, I hope that you can really appreciate this regular ordered structure of skeletal muscle. These structures have been called sarcomeres, and they extend from Z line to Z line. As you can see in this particular electron micrograph, we've shown you where the boundaries of the sarcomere are located. And it's important for the next few mini lectures to keep an eye on those Z lines and to watch what happens to the Z lines during muscle contraction, because that's going to help us understand the molecular events that take place that are responsible for force production and shortening of skeletal muscle. So here's an illustration. Oftentimes, it's really nice to see the raw data, the actual electron micrograph, and then compare it with an illustration which can help you dissect out some of those structures. So below you'll see an electron micrograph showing you the Z lines, and then there's the striation shown in a longitudinal section, and then there's also a transverse sectional view, so you can see the thick and the thin filaments. So this slide is showing and conveying to you that the sarcomere is this repeated structural unit in skeletal muscle. The sarcomere can be broken down into other components. One is the M line, which occurs right in the middle of this sarcomere. The other is the H zone and the A band. So these are structures that scientists have been able to label just based on their density in electron micrographs. And what's also important to convey at this moment is that the sarcomeres contain all of the components that are needed for skeletal muscle contraction. So scientists at the time observed these thick and thin filaments, and they knew that skeletal muscle shortened as it contracted. And so that led to the hypothesis that the thick or thin filaments themselves were altered in length during contraction that led to the shortening of skeletal muscle. So they wanted to test that hypothesis, and to do that, they conducted experiments where they examined the length of the thick and the thin filaments before and after contraction. So in a relaxed state and in a contracted state. And the tool that they used to do that was electron microscopy, which we've seen so far. Now, here is the data. As you can see in the top image, we're looking at a relaxed skeletal muscle. Identify the Z bands for yourself. Notice where they are. Identify the A band. Identify the I band. Now look at the image below, which is the contracted skeletal muscle. What happened to those Z lines? What happened to the thick and the thin filaments? Well, after taking several careful measurements, scientists found that those thick and thin filaments actually overlap as skeletal muscle contracts. So, here's an illustration showing you the sarcomere in a relaxed state. Again, notice where the Z lines are. Look at the thick filaments in the middle of the sarcomere. Look at those thin filaments in blue. What happens to them during contraction? This is what happens to them during contraction. You can see that those Z lines moved closer together, but the actual overall length of the thick filament and the overall length of the thin filament didn't change. It's the overlap that changed during contraction, and it's that overlap that allows the skeletal muscle to shorten as it contracts. And so what I just showed you or told you about the different changes that took place in the sarcomere are articulated below. And so that led scientists to the conclusion that, hey, there isn't any change in the actual length of the thick and the thin filaments that's occurring during contraction. It's this whole concept that they're overlapping with one another, and that overlapping is allowing for skeletal muscle to shorten during contraction. And that was actually a major finding back when skeletal muscle physiology was just being investigated. So that led scientists in the early 50s to generate a sliding filament model to explain muscle contraction. And this was championed by Hugh Huxley, who is one of the grandfathers of skeletal muscle. So let's get back to our case study. Remember, we were talking about the neurologist who was seeing several patients that she suspected had an inherited muscular dystrophy, and she sent the biopsies away for analysis by electron microscopy. Now, you've seen many examples of normal electron micrographs. Look at these again. What do you see? Well, they certainly look like transverse sections. You can certainly spot areas in these sections that look fairly normal. You can see the Z lines and make out sarcomeric structures. But there are many areas that are blurred, suggesting that the structure is completely disrupted. Already from what we know about skeletal muscle physiology, you can speculate that that blurred structure, that the disorganization that is so evident in these biopsies will impair skeletal muscle function. And indeed, each of these patients was diagnosed with a limb-girdle muscular dystrophy. In particular, this limb-girdle muscular dystrophy is caused by mutations in a gene encoding a protein that is at the Z line. So that blurring is called Z-line streaming, and it's very common in these types of limb-girdle muscular dystrophy. So I hope you can see that the structural arrangements, both within the sarcomere and the interaction of the skeletal muscle fiber with its surrounding layers of connective tissue, are really critical for skeletal muscle function. So that wraps up our first mini lecture, and I look forward to seeing you in the second mini lecture.